Part 2 of Chapter 4 mainly deals with how proteins are made inside of our cells. First is just talking about how different food molecules can be turned into ATP. Here it says carbohydrate molecules can enter the catabolic pathway, to make energy, or anabolic pathway for storage. Now here it says that too much glucose, if it's not being used, it gets converted into glycogen, which just means a chain of many glucose molecules just attached together. So then this is stored in our liver and muscle cells, but also too much glucose can get converted into fat. Alright, now we'll go into how proteins are made. So first, DNA. DNA is what is being used as the code or the instruction in making different proteins. Both DNA and RNA are made up of nucleotides. So here this picture is showing a nucleotide. Imagine many of these stacked on top of each other. If you have 2 rows of these, you get DNA, but if there's only 1 row, then it's RNA. Okay, so a nucleotide includes a sugar part, phosphate, and the base. Because in DNA, 2 strands get together, in the inside part of the DNA, the bases are attaching together. Here inside, the C and the G go together, and the A and the T go together. We can see on the picture here how in the DNA, the bases on the inside are attaching. G's and C's are attaching together, and the T's and the A's go together. Hydrogen bonds hold these bases together, but here once again, it's written here, bases pair specifically. This is called complementary base pairing. Alright, but now let's go into protein synthesis. It involves 2 steps, transcription and translation. In this first step, transcription, here we're using the DNA to make mRNA. So really no transcription is just making mRNA, forming mRNA. The enzyme that is forming mRNA is called RNA polymerase. Alright, so mRNA is made in this first step. Then in the next step, step 2, translation. Here, that mRNA that was made is going to get used, but we're also going to see these little smaller molecules, tRNAs, those get used, and the ribosomes to make proteins. Let's start with transcription. Here during transcription, the DNA is opened up by this enzyme called RNA polymerase. So here this pink oval shape, that's actually an enzyme that sits on the DNA and opens up its 2 strands. And so now those bases are exposed. Using only 1 strand of the DNA, then this RNA polymerase grabs free RNA nucleotides. So these are individual RNA bases. It grabs them, attaches to the DNA bases just temporarily, and attaches these pieces together. And so here, this red part that is forming, that's the mRNA. Once the mRNA gets finished being made, once the RNA polymerase gets to the end of the gene, the DNA segment, then the DNA closes back up and the mRNA is going to leave the nucleus. Now do know that when mRNA is made, so the individual pieces of the RNA nucleotides that are grabbed, it depends on what bases are on the DNA versus the mRNA. So for example, in the open part of the DNA, if there's an A base, for example, let's say this part was the A, the RNA piece that's grabbed and attached here temporarily is going to have the uracil base. Right? So if there's an A, mRNA that is made turns to U. If there's a C on the DNA, then it turns to G, G to C, T to A. And so here, if we just kind of write down these rules, DNA to mRNA, if there's an A on the DNA, that turns to U. If there's a C, then it turns to G, G to C, and if there's a T on the DNA, that turns to an A. There will be a question on the exam if I give you the DNA bases, can you write down what the bases would be for the mRNA that is being made? And the second step is translation. Now we're going to use that mRNA that was made, and this step has to use ribosomes and tRNAs to put amino acids together. This occurs in the cytoplasm, but know that ribosomes are used during this process. So here, mRNA leaves the nucleus, attaches to a smaller piece of the ribosome first, then a larger piece attaches. And so then, on the mRNA, each amino acid we're going to see is specified by a 3-base sequence in the mRNA called the codon. Each 3-base sequence in mRNA and DNA is a codon, so I'll explain this better on the next slide. I'll draw a picture. Then we're going to see these smaller molecules called tRNAs. tRNAs are going to carry over an amino acid. And at the bottom part of the tRNA is called an anticodon. And here we're going to see 3 bases in the tRNA. Alright, so let me show you what's happening in translation. Then I'll show you pictures that are from our book as well. Now let's pretend here that this is mRNA. Alright, so on the mRNA, imagine the line, this is where the sugars and the phosphates are. So I'm going to put some bases here. Let's say this part of the mRNA has the A, this part has U, this part has G, C, G, A, U, C, A. Alright, so let's pretend it's the mRNA. So in the background, where the mRNA attaches is a ribosome. But now, around the ribosome, we also have these T-shaped molecules. And there are many of these, and these are called tRNAs. But really, the T stands for tRNA. At the upper part of the tRNA is an amino acid. At the bottom part of the tRNA are 3 bases UAC, for example. But again, there are many of these, and these bases can be different. So this part, these 3 bases, is called the anticodon. But on the mRNA, each 3 bases is called a codon. So here's a codon, the next part is a codon, next 3 bases, next one is a codon. Right. So imagine here we have the ribosome. And the ribosome has a docking station for the tRNA. Now this tRNA is going to go and sit on the mRNA because here its anticodon is complementary to the codon of this. And so imagine if we bring that tRNA here, it brings over its amino acid, and this one is UAC. Alright, so now we have that amino acid here. Well, so now the ribosome is going to move to the next part, to the next codon, and now another tRNA is going to attach. But now this one here, the tRNA that's going to attach here, so imagine the ribosome has moved, and now a new tRNA comes in. It's going to have a different amino acid based on what anticodon it has. But now for this to attach here, the anticodon of the tRNA must be G, C, U. Alright, then this new amino acid is going to get added to that previous one. So imagine many more tRNAs come over and bring over their amino acids. And so when these amino acids attach, like I said, you just get a polypeptide, which is just called a protein. This chart is showing what codons mean what amino acid is going to come over here. And so notice more than one codon can mean the same amino acid. AUG, this is where the translation is going to start, only where there's the AUG on the mRNA. This is where the first tRNA is going to attach. But once we get to UAA, UAG, or UGA, now that's where the translation stops. So no more tRNAs are going to come and attach. And so in the drawing that I made, this is what it's showing here. Except in this picture, not sure what happened, but this is missing the A on this tRNA. But yeah, so it's showing here. This part is the mRNA. This one is the tRNA. It brings over an amino acid. The 3 bases at the bottom, it's called the anticodon. But yeah, so we always start at AUG. Now there might be bases before the AUG here on the mRNA, but no tRNAs are able to come and attach there. Only when we get to AUG, that's when we start. Okay, and now you can see more tRNAs coming over, bringing their amino acids. And on this next picture, again, it's just showing the elongation, meaning extension of the polypeptide. And so here, these circular shapes, those are the individual amino acids that were attached together to make up the protein or the polypeptide. There are 20 different types of amino acids, 64 possible codons, meaning that more than one codon translates to an amino acid, the same amino acid. Start codon is AUG, and then there are the stop codons, where we stop translating. In human DNA, we share 99.9% of DNA among humans, but the difference among people, well, that's gonna then changes the way we look, affect our health, affect our appearance. And next, we'll talk a little bit about mutations. Mutations are changes in the DNA sequence. Mutations can occur when bases are changed, added, or deleted. Now, this can happen on its own. For example, spontaneous mutations. So this can happen when an insertion of an unstable base occurs. So that would be spontaneous versus induced. Now, this would be due to exposure to mutagens. Mutagens are chemicals or radiation that cause mutation. So if you look on the picture here, on the DNA, it should be TTC, but here that second base got changed to A. And so that's a mutation. So no longer is the amino acid for that codon glutamic acid, but now it gets changed to valine. Some mutations are not harmful. Some mutations might be good. So here, in the Duchenne muscular dystrophy, this results in mutation in the gene coding for dystrophin, and so this would be the bad kind of mutation, because here the protein that's being made ends up getting changed because of that mutation. Now that single letter, single base change can lead to a big impact. But sometimes some mutations though are good. For example, some people have this mutation that protects them against HIV. That's because here the protein that gets made because of this mutation, protein is different. This is the protein on the surface of the cell. So this is where the HIV virus should be attaching, but it's not able to bind to it because the protein here has become different. Thankfully, we have these special enzymes called repair enzymes that repair, fix mutations. And so these mutations occur regularly, But yeah, so these enzymes go and check the DNA to see, hey, if any mutation occurred. Then they cut out the mutation and replace it with the correct bases. Now, again, because more than one codon code for the same amino acid, sometimes mutations, when they occur, it may not lead to a big result, big change, since it might code for the same amino acid still, even though there is the mutation. Also, having 2 copies of each chromosome, because we have our mom and dad chromosomes in our cells. If one copy is mutated, then the other copy, whether it's maternal or paternal, may provide enough of the normal function.